We have used 5 sources of experimental evidence to assess gene expression in the rice genome. This includes ESTs, full-length cDNAs, MPSS, SAGE, and proteomic data. Genes that have expression support are annotated as express protein or amended with expressed. From the homepage, we provide 3 major tools to analyze gene expression in the rice genome. These are the Rice Genome Expression Anatomy Viewer and Digital Northern tool, the Highly Expressed Gene tool, and the Gene Expression Evidence Search tool. In the Rice Gene Expression Anatomy Viewer and Digital Northern tool, we provide digital northern data from ESTs to assess expression of loci through rice tissues. A locus or list of loci can be inserted in the box, or alternatively, a file listing loci can be uploaded. Using the sample locus given, we can select Submit and see that this locus has 53 mapped ESTs and full-length cDNAs. This can be viewed through the Anatomy Viewer or the Digital Northern Viewer. The Anatomy Viewer provides a graphic representation of EST frequency using a color code that represents the frequency of ESTs detected. For this locus, 51 ESTs were mapped. Note that the full-length cDNAs are not included in this analysis. The columns can be sorted by clicking on the header line. Let's click on the number of ESTs found to get the tissue with the highest number of ESTs on top. The columns represent tissue source for the EST that is hyperlinked to a list of cDNA libraries constructed from leaf tissue that were used to generate ESTs. The library ID is linked to an EST library and tissue report page that has the library name, cultivar, tissue type, and the description of the library as provided by GenBank. If we go back, the plant ontology for the tissue is hyperlinked to the plant ontology website. The anatomy view shows the color shading based on EST frequency. Also listed are the total number of ESTs from the tissue, the number of ESTs from the tissue type that map to this locus, and the frequency of ESTs for this locus in the given tissue. If we go back, we can click on the Digital Northern link, and from there, each cDNA library with an EST that was mapped to this locus is shown. The total number of ESTs from each library is provided, along with the number of ESTs mapped to this locus and the calculated frequency of representation of this transcript in the cDNA population. The library ID is hyperlinked to the EST library and tissue report page that provides the library origin. The tissue is hyperlinked to the EST library and tissue report page that shows all the EST libraries derived from the tissue.